Polysaccharides are a diverse set of molecules that play important roles in biology due to their frequent presence on the surface of cells. In most microbiological contexts, the cell surface is the only thing that other cells and molecules can see, since the inner contents of the cell are secluded by the membrane. Despite their importance, their biosynthesis is far less studied and engineered than other types of metabolites. In this paper from Feldman, a chimeric biosynthetic pathway between a surface polysaccharide and a glycoprotein lead to an interesting example of composability. Carbohydrates are particularly useful as therapeutics. Small molecule carbohydrates are a common class of secondary metabolites, especially as antibiotics. For example, Optimer Pharmaceuticals is a pharmaceutical company founded on the idea of combinatorial small molecule carbohydrate drug discovery. Additionally, carbohydrates are sometimes necessary for the function of therapeutic proteins, or if introduced synthetically, could improve the serum half-life or immunogenicity of a therapeutic protein. For all carbohydrate engineering, there are soluble nucleotide sugar intermediates. The enterobacteria produce a diverse array of capsular polysaccharides, and as a result, there are enterobacterial pathways for many nucleotide sugars that could be easily moved to an engineered E. coli. Here's an example of one of these pathways that takes glucose 6-phosphate to UDP-GLICNAC. So you really only need to add transferases and polymerases to design new carbohydrates. The nucleotide sugars are off-the-shelf pathways. In this paper, the authors show you can make a chimeric biosynthetic pathway between two quite different polysaccharide biosynthetic processes. In this study, the authors are splicing together two pathways that both involve an oligosaccharide. In the N-glycosylation biosynthesis pathway from C. jejuni at left, monosaccharides are iteratively added to a growing chain by glycosyl transferases. The resulting 7-member oligosaccharide is linked to the inner membrane by a lipid linker and then transferred to the periplasm, where it is transferred to a protein called ACRA. This results in a glycoprotein. The O-antigen biosynthesis in E. coli similarly starts with the lipid-linked oligosaccharide that is transported across the membrane, but then is polymerized into a 40-unit polymer and transferred to another lipid anchor. These two pathways involve different oligosaccharides, but beyond this consideration, they perform the same sequence of steps to give rise to this periplasmic oligosaccharide intermediate. What they show in this paper is that they can splice the two pathways together over this intermediate and obtain glycoproteins in which ACRA has picked up the O-antigen's oligosaccharide. Predicting that this sort of composition should work is challenging. It all boils down to the substrate specificity of the particular enzymes involved in transferring and polymerizing these oligosaccharides. Sure, superficially this chemical intermediate looks really similar between the two systems. It has a bunch of sugars and they are attached to the same lipid linker. However, they employed quite different sugars in different configurations, and it is unclear which of these enzymes will tolerate which substrates. We don't currently capture models of substrate specificity at all in biochemical databases. This is particularly true of these more exotic enzymes.